Howdy, folks. Welcome to the Chapter 9 notes on DNA. This is the first topic of Unit 5. Molecular genetics. In the first part of this notes, we're going to talk about DNA structure and how it reflects its role as the genetic material. The rest of the notes will come later. First, you need to understand that before the discovery of DNA as a genetic material, there was only evidence to suggest that something was being inherited or passed on from parent to offspring. And there was some evidence beginning to accumulate that DNA was that genetic material. So it was found that DNA could be shown inside cells by staining it. And that's where the name chromosome comes from. Chroma means color, some means body. So when they were discovered, they were called colored bodies, or in other words, chromosomes. Now, the discovery of nucleic acids had already taken place. Nucleic acids were shown to be made of CHONP, and there was just evidence. There wasn't much evidence to support it as the genetic material, although there was some, such as, for example, after the S phase, the amount of nucleic acid material doubled, but there was still a really strong contender for proteins. Proteins were believed to be the genetic material because they're so diverse, because they have so many functions, and they could be created with different sequences of amino acids to be as highly reflective as the amount of diversity in life. So there was a big debate whether or not proteins or nucleic acids were the genetic material. Now, Hershey and Chase were able to definitively close that book and end that argument and prove that DNA, not proteins, was the genetic material. And the way that they did that is they used viruses that are called bacteriophage. And bacteriophage are viruses that specifically target bacteria. Now, they use these because they do contain DNA and proteins. In their experiment, they showed that when a virus infects a bacterium, it injects only its DNA. So the experiment we're talking about here is the one from your flipped assignment, your Hershey and Chase. And what Hershey and Chase were ultimately able to prove was that it was the DNA, not the proteins, that entered the cell or entered the bacteria and therefore was the genetic information. So all the focus now was on DNA, and there was a race to discover its structure. Now, DNA was known to be a polymer of nucleotide monomers, but its three-dimensional structure was still unknown. Think back to Unit 1, where we talked about the structure of this nucleotide and the three components that create it. Now, once they understood the structure, scientists would be able to understand how it replicates. Now, the structure was deciphered when many experimental evidence were considered together as a whole. The first of which was done by a scientist named Rosalind Franklin, and she used X-ray crystallography to figure out what the shape of that DNA was. And so she took what are known as crystallographs. Now, you don't need to know the specifics about how X-ray crystallography works. You just need to know that Rosalind Franklin took these crystallographs, which are made from X-ray crystallography, and created a picture that showed or suggested that spiral or helical model. So this is just a diagram showing you how it works. Again, you don't need to worry about the details of how X-ray crystallography works. But this right here is the image we wanted to show you. This right here is the picture that was taken by Rosalind Franklin. So this is the picture, and this is what is suggesting that helical model. Now, scientists knew the chemical composition of nucleic acids at this point. They knew that nucleotides were consisted of that sugar deoxyribose, that phosphate group, and the nitrogenous base. And the nitrogenous bases were divided into purines or pyrimidines. Purines were adenine and guanine, pyrimidines were cytosine and thymine. All of this we talked about back in Unit 1. We even mentioned how Chargaff had created or had found that the proportion of A's was always equal to T, and the proportion of C was always equal to the amount of G. So, in other words, they complement each other, they pair up. Now, one of the things we also mentioned back in Unit 1 was the hydrogen bonds. And 2 hydrogen bonds form between the adenine and thymine. Between the cytosine and guanine, there's actually 3 hydrogen bonds. And remember, hydrogen bonds are weak interactions. However, when you have many of them, millions of them, holding the DNA strand together, they are considered a strong force. It was Watson and Crick that combined all the knowledge of DNA to determine its structure. So they took information from Franklin and from Chargaff and from other scientists, and they put the puzzle pieces together. They did do some measurements that suggested that the 2 polynucleotide chain, that there are 2 chains in the model, and that the model showed that the DNA strands must be anti-parallel. In other words, what they did was take other people's work and put it together into one final puzzle. So what you see on this diagram were the 2 kind of competing models at the time, where it was thought that maybe the phosphate sugar backbone, which is shown right here, was pointing towards the inside of the DNA and not the other way around. Now, Watson and Crick were able to discover that model and found that the sugar phosphate backbone is on the outside, and that the nitrogenous bases were the ones that faced towards the interior. They were also able to use Chargaff's rules to look at the pairing itself. So adenine pairs with thymine, not adenine with guanine, cytosine with thymine. So let's talk about the structure of the DNA. DNA is double-stranded helix of uniform diameter. The chains are held together by hydrogen bonds between the base pairs and by van der Waals forces between adjacent bases on the same strand. We had mentioned already that 2 form between adenine and thymine, and 3 hydrogen bonds form between cytosine and guanine. The 2 strands are anti-parallel, and anti-parallel is referring to that sugar phosphate backbone, which we talked about back in Unit 1. So keep in mind that when you're looking at a sugar or you're looking at a molecule, this is, for example, deoxyribose, and from the oxygen group, the first carbon we encounter is carbon number 1. And from there, we count clockwise. So this is carbon number 1, number 2, number 3, number 4, number 5. So this is a pentose. Now, if this were ribose, we would see an additional hydrogen group or additional hydroxyl group attached here at carbon number 2. So you would see an OH, but you can see that it doesn't have the OH group. The oxygen has been removed. Therefore, this is called deoxyribose. So when we're talking about the anti-parallel strands, in other words, the 3' to 5' end is opposite the 5' to 3' end in that double helix. What these numbers here are referring to is which carbon is going to get, or which carbon is the carbon we are encountering. So if you were looking at it from this direction, starting up here, as you go down, the first carbon you encounter is carbon number 5 on this side, on the backbone side. So the first carbon you encounter is carbon number 5. So this end, or this strand right here, would run 5' to 3'. If this model were upside down, then it would be 3' to 5'. So you can see here a larger picture of the strands. And so it's very important to look at this from the point of view that they are referring to. So on this one, it's starting down here at the 5' end. So here's this 5' carbon. So here's your oxygen. This deoxyribose is upside down. So this is the oxygen. This is carbon. This is carbon number 1, number 2, number 3, number 4, and then that's carbon number 5. Again, because this deoxyribose is upside down. So from this point of view, going this way, this is the 5' carbon, and then the last one you would encounter is the 3' carbon. On the other side, it's probably a little easier to see it on this side. So here is my oxygen group, and then so from here, we're going to count. So this is your 5' end, so this is 1, 2, 3, 4 in that corner, and then this is number 5. So when you're going in this direction, this is the 5' end. So this is the 5' to 3-strand end going in this direction. So these two strands, one of them is going to be upside down compared to the other. So the surfaces of those nitrogenous bases are chemically distinct. And what's going to happen is you're going to have the binding of proteins to the specific base pair sequences. And that's very important for DNA-protein interactions. Remember that proteins can act as enzymes, and we're going to talk about several enzymes. So let's say that you have a strand of DNA here as shown. Very, very simplified. So this is the sugar phosphate backbone, a very, very simplified version. And then these are the nitrogenous bases that are sticking up out of the These are the nitrogenous bases that are sticking out of the DNA. So notice the shape, the overall shape of those nitrogenous bases. And so when you look at the shape here, the overall shape that is formed by these nitrogenous bases, try to picture an enzyme that would fit in this groove right here. So the enzyme active site would need to be specific to this shape. Let's see what that would look like. Here is another very simplified diagram of an enzyme, and it's got the right active site to fit with the bumps and curves here of the DNA. This means that enzymes can have active sites that are very specific for regions of the DNA. And this is very important in the process for replication, which we'll talk about next, and gene expression, which is chapter 10. So in summary, DNA structure is essential to its functions. It is the storage of genetic information, and its precise replication during the cell division is because of the complementary base pairing. There are things we're going to talk about, such as mutations that would change the stable structure of that DNA. And then we're also going to talk about how the expression of coded information is what's known as the phenotype. In other words, that the proteins end up being the phenotypes. Thanks so much for watching, folks. Bye.